Here at the ISB, we do lots and lots of patient studies, sometimes involving hundreds of patients. These patients may be cancer patients, it may be patients with COVID, it may be patients with MS. We want to understand in great depth about those patients' diseases and how best to treat them. And in order to do that, we need to look at the patient's blood. And this video is to tell you about how we develop a quality biobank for all of those patient studies that we do. When you sign up for a patient study, you'll check a little box that says yes. And once you check that box, lots and lots of things move into motion. And when you go and you give your blood, we're going to take that blood and we're going to end up putting it into lots and lots of little vials that contain all the components of the blood, from cells to plasma to serum to whole blood, etc. And out of that whole blood, we're going to measure your genome, we're going to measure your transcriptome, we're going to measure your proteome, we're going to measure your metabolome, we're going to measure your PBMCs, we're going to look at the T cell diversity, the B cell diversity, and we're going to look at other biospecimens as well, such as the microbiome. But today, I want to talk to you about how do we ensure that when we do those measurements, they're high quality. And ultimately, that means that the biobank from which those specimens come has to be of pristine quality. And something that we pride ourselves on at the ISB is that our biobank is basically second to none. And in order to make a quality biobank, we want to go as fast as we can from the blood in the patient's arm to our cryo storage facility here with all the blood processing steps happening in between. To make that happen, there's several steps. First, we get called when we're told that a patient is coming in to get a blood draw. And then we have operators standing by to take the blood that's drawn in the clinic, and immediately that blood is couriered to the ISB. Then that blood goes into a blood processing facility. There's several processing steps. We have to centrifuge the blood, we have to separate the plasma, we have to separate the cells. A critical aspect of this is that if you don't do this quickly, all of the molecules and all the cells that we care about will degrade. And in fact, we want those molecules to be intact, and we want those cells to be alive when we study them later. So if you're a patient who checks a box to participate in one of our studies, checking that box puts into motion about 12 people who are going to work rapidly to make sure that your blood is processed and biobanked in the highest possible quality to allow us to pull as much information as we can to help understand your disease and to help develop new treatments for your disease.